Bismillahirrahmanirrahim. Today we will talk about the modes of inheritance. Traits are transmitted from parents to their progeny by gametes. The 23 chromosomes of a gamete carry the estimated 30,000 genes that express the human traits. Each chromosome carries hundreds to thousands of these genes. These genes are transmitted from parents to children during fertilization. In fertilization, sperm nucleus enters the egg to fuse with its nucleus and form the zygote. The zygote has a diploid nucleus containing two genomes represented by 23 pairs of homologous chromosomes. Each one of these pairs has a paternal and a maternal homologue or chromosome. The paternal and maternal chromosomes carry the gene at certain loci. For example, chromosome 15 carries the eye color gene at this locus. The gene may be expressed in two or more versions. Each of these versions is called an allele. For example, the eye color may be brown or blue. The brown is a dominant allele and expressed by capital B, and the blue is the recessive allele and expressed by small b. Now, the allele expresses different varieties of the same trait. So this is brown and this is blue. Both of them are for the eye color. Modes of Mendelian inheritance. We know that the chromosomes are either autosomes or sex chromosomes, which is XY. The pattern of transmission of autosomal genes is called the autosomal inheritance. Dominant trait is expressed when even a single chromosome carries the allele in both males and females. However, the recessive trait has to be expressed on both genes in both chromosomes, and so both chromosomes should carry the allele. Now, if one chromosome carries the dominant and the other carries the recessive, this is called a carrier. The sex-linked or pattern of transmission of genes is usually on X chromosome, which carries most of the sex-linked genes. If dominant trait is expressed when a single chromosome carries the allele in female, although she has two chromosomes, but when just one of the X chromosomes carries the dominant allele, she will show the dominant characteristics. In male, because we have just one X chromosome, when it carries the allele, this allele will be expressed. However, in recessive trait, X-linked trait is expressed in female only when both chromosomes carry the allele. But in male, only one allele expresses the trait. Why? Because it is the only gene found in the male, on the X chromosome of the male. In female, if one chromosome, X chromosome, carries the dominant and the other carries the recessive, this is a carrier. Because the male has only one X chromosome, and so he carries just one allele on the sex chromosomes, he is called half zygous pair, half a pair, hemizygous. However, in the case that both chromosomes carry the same allele, this is called homozygous. If one chromosome carries the dominant and the other recessive, this is called heterozygous. Some cases there are more than one allele controlling the trait, such as the ABO blood group. The ABO system in the humans is controlled by three alleles, usually referred to as IA, IB, and IO, and they are located on chromosome 9. I stands for the production of isohemagglutinin molecule, which appears on the surface of RBCs. IA and IB are codominant, so even if they are found together, it will form AB. But if one of them is found alone, it will appear, as we will see in a while. Type A and type B antigens are found so that we can, if we look at the blood type A, the isohemagglutinin migrates to the surface of RBCs, the isohemagglutinin A. In this case, the serum will contain anti-B, not anti-A. Otherwise, if there is anti-A, it will be clotted. So only anti-B. That's why if the cell from B enters here, they will be clotted. In the case of blood type B, the isohemagglutinin on the surface of the RBCs is B, isohemagglutinin B, and the serum contains anti-A antibodies. In the case of AB blood group, we'll find that both isohemagglutinin A and isohemagglutinin B are found on the surface of the RBCs, and the serum does not contain any antibody for the A and B isohemagglutinin. In case of blood type O, the red cells has no isoglutenin on their surface, and in this case the serum contains both anti-A and anti-B antibodies. Now, genetically, if we look to the genotype of the person and his blood group, we'll find that in the case of IA, that is paternal chromosome has express the A isohemagglutinin, and the maternal expresses also the A isohemagglutinin. This person will be blood group A. If it is IAIO, it will be also A. If it is IBIB, it is B. If it is IBIO, it is also B, because B is the dominant. But if it is IAIB, this is codominant, and so this will be AB blood group. If it is IOIO, no isohemagglutinin at all, and the blood group is O. Now, in order to get information about a trait or disease that appears in a family member, we have to draw a pedigree. By analyzing a pedigree, we can determine the mode of inheritance and predict how a trait will be passed on. The symbols for drawing a pedigree are internationally used. The square in the male is represented by a square and a female is represented by a circle. If it is open, it's unaffected, but if the circle or square are filled, this is an affected person. The presence of an arrow on the circle or square means that this is the first person to be represented or presented to you as having the trait or the disease. And this is called the propositus or proband. If the circle half-filled circle or half-filled square, this is a carrier, heterozygous carrier. In the case of X-linked female who has two X's, one of them having the dominant and the other the recessive, and so she is a carrier. She has a circle, she is represented by a circle with a dot in the middle, and this is gene carrier in the X-linked. The deceased person has a cross line on the circle or square, while the rhombus means that this is a sex. The sex here is unknown. Square with a like this is the without a person, a man without offspring, while in this case the this figure means the consanguineous marriage, that is the marriage between two relatives, and this means dizygous twin, while this means monozygous twin. Now, how do we draw a pedigree? We start with the first person, who is the propositus, and we write down all the information about him. Then his father, mother, and sisters and brother. Then we start to go up in the family tree, his father's mother and his father's father, and his aunt, her children. She has a boy who is normal, and we go to the mother, and the mother has her father and mother. She has a brother who has the disease, while she has a sister who has two of her sons free of the defect. The grandfather and the grandmother are dead. So, as we see, we start the pedigree from down there with the propositus or the proband or the first person that appears to us. Now, pedigree of different modes of inheritance. Autosomal inheritance. If we have a female who is heterozygous with dominant and recessive genes, these genes will be segregated during the formation of the egg cells by meiosis or meiotic division, and so some of the egg cells will have the dominant allele and others will have the recessive allele. If we have a man, he will also have spermatozoa which have the dominant allele and spermatozoa that have the recessive allele. Now, if they were married, their children here, the first child will take the dominant from the mother and the dominant from the father, while here this is a boy and he will take the dominant from the mother and the recessive from the father, and then this is, suppose it is a girl, she will get the recessive from the mother and the dominant from the father, while finally this one will take the rest, which is the recessive from father and recessive from mother. And so we will have homozygous dominant, heterozygous, and homozygous recessive, and there the ratio between them is 1 to 2 to 1. Let's take an example of a dominant, autosomal dominant trait, which is cleidocranial dysplasia. We'll have father and mother who are heterozygous, and they will have daughters, and as you see, the daughter will be homozygous dominant, taking dominant from the father and dominant from the mother. Then a boy who will take the dominant from the father and the recessive from the mother, and another girl who will take the dominant from the father and recessive from mother. Finally, the final one will take the two recessive genes, as you see. So we will have one homozygous dominant, two heterozygous, and one homozygous recessive. Anyone having just one dominant allele will show the trait. And so the phenotype, all of the three here will be dominant, while this will be recessive and normal. The ratio is 3 to 1. If we take the pedigree of a family, and how do we know that this family has, or the mode of inheritance of the gene in this family is autosomal dominant? We'll find that some of males and some females are carrying the trait in all generations, and both the males and females will represent the trait. So this is the autosomal inheritance. Now, if we take the recessive, for example, the dyskeratosis congenita, and we have father and mother who are heterozygous, their children will be, the first is homozygous dominant, the second is heterozygous, the third is heterozygous, and the last is homozygous recessive. Now, the dominant will not show the recessive trait. Only the one who have the homozygous recessive genes will show the trait. And so the phenotype, this will be dominant and normal, while this will be recessive and showing the trait, and the ratio is also 3 to 1. How do we know that this family is carrying an autosomal recessive trait, will find that both males and females will be affected, but not in all generations. It's sporadic. Some of them will show the trait. Allelic and non-allelic genes. Sometimes the gene is expressed by the same features in two different genes. For example, deafness can be caused by different genes at separate loci. Example A, which is on chromosome 3, and B, which is due to expression of a gene on chromosome 4. Now, anyone who marries one from this type A will have all his children having the homozygous recessive, and so they will appear deaf. But if someone from this family marries another one from the other family, their children will be heterozygous for both genes, A and B, and so their children will not be deaf. Why? Because the two are non-allelic. X-linked inheritance. X-linked inheritance, if we take a female who has X carrying, for let's say, the yellow chromosome, and the other the red chromosome, and a boy or a man who has the yellow chromosome and Y. Now, if they are married, then we'll find if the yellow allele in X and another yellow allele on the other X from male, this will be a female carrying homozygous yellow. But if the yellow allele of the female is with the Y of the male, this will be Y who carries the allele yellow. If X or the yellow X allele is with red X allele, this is a heterozygous female who has both alleles. Finally, if the red X allele is fertilized by Y allele, this will be a boy who has the red X allele. Now, if we take a case of X-linked dominant orofacial digital syndrome, a female who has the red, which is the syndrome, which can express the syndrome, and the black, which is normal. So she is heterozygous, and a male who has the normal X allele and Y. If we look at this family and say this family has 4 persons, 2 boys and 2 girls. Now, if a red X or affected X is with a black, this will be heterozygous female. But remember that this is dominant, so the presence of just one X which is red, will show the trait on this female. This male will have also the red and Y, and of course he will show the trait because he has just the X allele. This female will have both black X's, and so she will be normal. This male will have X and Y, and he will be normal. So the phenotype will show up that 2 will be affected and 2 will be normal, and the ratio is 1 to 1. Now, if we look at a family pedigree, how do we know that the mode of inheritance is X-linked dominant? In a family pedigree, we will find that if a male is affected, all females should be affected because they will take their X from the male. But if the female is affected, we'll find that both males and females of her progeny are affected. Let's take the X-linked recessive ectodermal dysplasia case, and we look at the family like we have seen in the last time, and here the red X means that it carries the recessive trait. Now, genotype will have black, normal, black Y, normal, black, and this is heterozygous, and as it is a recessive, it will not show the trait. But this boy will have the X, which is red, carrying the allele, the ectodermal dysplasia allele, and Y. So it is the expression will be only for the ectodermal dysplasia. And so we will find, although the phenotype of all of these is normal, this boy is affected. And you can expect that if we look at the pedigree of a family, we will find that the boys or the males are the only affected persons in this family. So this is X-linked recessive. Now, we said that the autosomes and sex chromosomes are found in the nucleus. And so what we have discussed before is the nuclear inheritance. But we know that the mitochondria contains in its matrix circular DNA. Although this circular DNA has genes, a small number of genes, which are expressed in the formation of tRNA and ribosomal RNA, and some of the genes necessary for formation of ATP, any mutation in these genes will affect the formation of ATP and the formation of energy. And so the tissues like the muscles and nerve tissues will be highly affected by mutations in the DNA of the mitochondria or mitochondrial genome. For example, mutation in the mitochondria of a person have been associated with diverse forms of human diseases and aging, such as progressive external ophthalmoplegia, as you see here. You see the eyes and the muscles of the eyes are affected, and This is a slow progressive paralysis of the external eye muscles. How do you know from looking at a pedigree that this family has mitochondrial inherited disease? If we look to how the mitochondrial inherited diseases are inherited, we'll find that during fertilization, the egg cell have the mitochondria which are transmitted to the zygote. And so the zygote who will form the cells of the body will carry only the mitochondria from the egg cell, but not from the spermatozoon. They will not enter the zygote. And so the mitochondrial diseases or the mutations that occur in the female or in the mother's mitochondria can be transmitted to her progeny, but the male does not transmit his mitochondria to the progeny. That's why we call this case the maternally inherited disorder. Now, let's look at pedigree and see how can we know that this is a mitochondrial inheritance. You see that if a female is carrying the disease, males and females, both males and females, will inherit the disorder. But if a male carries the disorder, none of his progeny will get the disorder, because the male gives spermatozoa, and the spermatozoa do not transfer their mitochondria to the next generation. And so when we look to the pedigree and find out that the females transmit the disease to both males and females, but the males do not transfer the disease to their progeny, we know that this is a mitochondrial inheritance. Thank you.